What is a teratogen? A teratogen is any agent that can disturb the development of an embryo or fetus, leading to congenital malformations or birth defects. For instance, exposure to certain mycotoxins like zearalenone during pregnancy can cause reproductive anomalies in livestock. Viral infections, pharmaceuticals such as thalidomide, or deficiencies like maternal iodine shortage can all act as teratogens. Teratology, the study of birth defects, helps veterinarians and toxicologists identify risks and recommend safe breeding practices. What is a carcinogen? A carcinogen is a substance or agent capable of causing cancer by inducing genetic mutations or promoting uncontrolled cell growth. For example, aflatoxin B1, produced by Aspergillus flavus, is a potent hepatocarcinogen in poultry and mammals. Chronic exposure to such carcinogens in contaminated feed can lead to liver cancer. Identifying carcinogens allows for risk reduction strategies, such as testing animal feed for contaminants and improving storage conditions to prevent mold growth. What is a mutagen? A mutagen is any physical, chemical, or biological agent that alters the DNA sequence, potentially leading to mutations that affect health. For example, benzopyrene, found in smoke from incomplete combustion, is a powerful mutagen that binds to DNA, distorting its structure. In laboratory settings, mutagens help researchers study gene function, but uncontrolled exposure can lead to cancer or inherited effects. Veterinary toxicologists monitor mutagen exposure to safeguard animal health and prevent reproductive losses. What is phototoxicity? Phototoxicity occurs when a substance becomes toxic upon exposure to light, usually ultraviolet, UV, radiation. For example, certain plants like St. John's wort, Hypericum perforatum, contain hypericin, which can sensitize the skin of grazing animals. When exposed to sunlight, these compounds cause severe dermatitis, known as photosensitization. Animals with light-colored or sparsely haired skin are especially vulnerable. Prevention includes avoiding photosensitizing plants and sheltering animals during high sunlight hours. What is synergism in toxicology? Synergism refers to an interaction where combined toxicants produce effects greater than the sum of their individual effects. For instance, simultaneous exposure to aflatoxin and acratoxin in poultry can lead to amplified liver and kidney damage, exceeding the effects seen when each toxin acts alone. Understanding synergism is critical in veterinary medicine, as animals are often exposed to multiple environmental toxins, feed contaminants, or drug residues at once. What is antagonism in toxicology? Antagonism is the interaction where one substance reduces the toxic effect of another, often utilized therapeutically. A prime example is the administration of atropine in organophosphate poisoning cases. Organophosphate inhibit acetylcholinesterase, leading to nerve overstimulation, while atropine blocks acetylcholine receptors, relieving the clinical signs. Recognizing antagonistic relationships allows veterinarians to employ effective antidotal therapies in toxic emergencies. What is selective toxicity? Selective toxicity describes a toxicant's ability to target one species or type of cell while sparing others. This principle is foundational in veterinary pharmacology. For example, antibiotics like penicillin target bacterial cell walls but do not harm animal cells, which lack such structures. In pest control, selective toxicity enables the development of rodenticides that exploit differences in metabolism between rodents and other mammals, minimizing collateral damage when used responsibly. What is idiosyncratic reaction? An idiosyncratic reaction is an unpredictable, atypical response to a toxicant or drug, unrelated to dose or exposure duration. These reactions often stem from genetic peculiarities in metabolism or immune function. For example, certain dogs exhibit idiosyncratic liver failure when exposed to carprofen, a commonly used NSAID, despite normal dosing. Since these reactions are not dose-dependent, they pose diagnostic challenges and necessitate vigilance in post-treatment monitoring. What is tolerance in toxicology? Tolerance refers to the progressive decrease in response to a toxicant following repeated exposure, requiring higher doses to elicit the same effect. For example, ruminants exposed to gradual increases in dietary nitrate may develop tolerance by enhancing their ruminal microbial capacity to reduce nitrate to harmless nitrogen gas. However, sudden exposure to high nitrate levels can still overwhelm this adaptation, leading to toxicity. Understanding tolerance mechanisms helps veterinarians manage exposure risks in livestock. What is detoxification in toxicology? Detoxification is the biological process of converting toxic substances into less harmful or excretable forms. This typically involves liver enzymes transforming lipophilic toxins into water-soluble compounds, which are then excreted via urine or bile. For example, cytochrome P450 enzymes play a pivotal role in metabolizing xenobiotics. Detoxification is central to both natural defense mechanisms and therapeutic interventions, such as using activated charcoal to absorb ingested toxins and facilitate their elimination.